Members of the Presidency, authorities, distinguished guests. On behalf of my colleagues Yossi Schlesinger and Charles Sawyers, I am deeply honoured to accept the BBVA Foundation Frontiers of Knowledge Award in Biomedicine, which is being given for our work on tyrosine phosphorylation and its role in cell signalling and the development of drugs that target tyrosine kinases for cancer therapy. I'm particularly grateful to be here because when I first learned of the award, I was lying in a hospital bed recovering from a serious bicycle accident. Like many discoveries, there was an element of serendipity in the way that I stumbled on tyrosine phosphorylation. At that time, in 1979, we were studying a DNA tumor virus transforming protein and had just shown that it has a kinase activity that adds phosphate to proteins. I was doing the routine test to determine the amino acid our enzyme attached phosphate to, expecting serine or threonine, the phosphoamino acids known at the time. This meant breaking up the protein into single amino acids and separating them on a plate by applying an electric current. The night I did the experiment, I was too lazy to make up a fresh lot of the necessary buffer solution. And because I used an old solution, a novel phosphorylated amino acid was revealed. The mystery compound proved to be phosphotyrosine, and a new type of protein kinase was born. Yossi Schlesinger began working on the epidermal growth factor receptor tyrosine kinase in the 1970s, and his subsequent work has revealed how EGF binding to the receptor transmits signals inside the cell. Once it emerged that activated tyrosine kinases can cause cancer, he founded a company to make small molecule tyrosine kinase inhibitors for cancer therapy. His company, Sugen, successfully developed a number of inhibitors that target cancer-causing tyrosine kinases, including Sutent, an FDA-approved cancer drug. Charles Sawyers began working on chronic myelogenous leukemia and the role of the BCR-ABL tyrosine kinase in this disease 25 years ago. He realized that targeting the BCR-ABL kinase activity could become a new type of CML therapy and led clinical trials that resulted in the approval of Gleevec, a small molecule inhibitor of the BCR-ABL kinase, as a new and very effective treatment for CML approved by the FDA in 2001. Gleevec became the harbinger of what is now a large number of tyrosine kinase inhibitor drugs in use for cancer therapy. It goes without saying that many people in our groups and other scientists around the world, as well as our families, have played a key part in the research that led to the discovery of tyrosine kinases and the generation of drugs that target tyrosine kinases, and we owe them all a debt of gratitude. It is indeed very satisfying that a discovery made over 35 years ago has led to the development of a treatment for a devastating human disease. Finally, let me acknowledge the BBVA Foundation and thank them for their hospitality and the honor that they have bestowed upon us with the Frontiers Award in Biomedicine. Thank you.